I'm Daniel Levine, and this is the Bio Report. When Takeda in 2023 paid Nimbus Therapeutics $4 billion upfront and the potential for 2 additional $1 billion milestone payments for its experimental TYK2 inhibitor, the deal was an eye-popping validation of Nimbus's approach. The company, an early innovator in computational chemistry, has now integrated AI into its approach to drug discovery. And though it's been an innovator in technology, it's also been an innovator in its portfolio approach as an example of an effective use of a hub-and-spoke business model. We spoke to Abbas Kazimi, CEO of Nimbus, about computational chemistry, how the company's drug discovery approach has evolved with AI, and how its business model provides liquidity to investors while making it easier to structure deals. Abbas, thanks for joining us.
Daniel, what a pleasure to be here. Thank you.
We're going to talk about computational chemistry and drug discovery, Nimbus Therapeutics, and to what extent AI has changed what Nimbus is doing. Let's start with the founding of Nimbus in 2009. It was an early innovator in the use of computational chemistry. Perhaps we should start there. In 2009 terms, what was meant by computational chemistry?
Again, thank you so much, Daniel. Always a pleasure to talk about what Nimbus has led in the world of computational chemistry and still continues to advance those tools that we've had and really think them through how to continue to utilize them to best of our abilities to make molecules that matter. In 2009, we were founded on the principle that computational chemistry had existed for a while. It was around, Schrodinger was well present, advancing these tools and software and applications for a number of years. And as Nimbus was getting put together, it was based on a couple of principles. The first principle was, can we create a novel business structure, which we'll talk about later today, that can be capital efficient, that can really focus on advancing assets and not fundamentally validating a platform. Number two, kind of a more virtualization of the organization. So as CROs were becoming more capable in terms of talent and offerings, could we create a business that allowed to leverage, universally across the globe, a different set of talent? And then to your specific question, computational chemistry was primarily referring to more physics-based modeling, and Nimbus's specialty was very specific around structure-based drug design to drug discovery, which ultimately meant, can you have computer-driven simulations to understand and predict how drug molecules interact with biological targets? Then we were kind of guiding by our work on resolving structural data. And so over the past 15, 16 years, we have developed a number of relationships to extract and understand the structures of these proteins that we're going after. And that's going from using fundamentally X-ray crystallography to some of the Nobel Prize winning achievements across cryo-EM. And these techniques, in 2009, they weren't really mainstream part of drug discovery at most biotechs or pharma. They were kind of recognized as promising, but mostly limited to some of these more experimental groups. And so for us, Nimbus was kind of the early pioneer of taking what people were using as force extenders outside their organization and really bringing them into the heart of the business model. And so that was Schrodinger coming together with Atlas Ventures of saying, let's put this into a newly formulated drug discovery company that has novel targets, and novel could be debated on what novelty really means, and then really think about building a biotech by taking these computational tools and this computational chemistry as the core engine and expanding that further out.
And what problem was using this type of computational chemistry approach seeking to solve in drug discovery?
It's probably more fundamental around some of the challenges that more predictive properties. A lot of it was kind of getting into more in silico prediction. So getting away from some of the brute force, um, you know, screening methodology that was available, getting into more virtual screening, getting into understanding some more of the tools around molecular docking, right? Could you actually simulate the binding of small molecules to their target proteins, predicting the optimal binding modes and affinities? Molecular dynamics, that's a physics-based approach to stimulate movements and flexibility of proteins. And so I think these tools were available, Daniel, even using some of the more popular, and I would say still probably the best tool out there that many biotechs and pharma still uses, is using some of these free energy calculations. Again, really estimating binding affinities. It was looking at the targets that we had that no one was making progress against because of the chemical properties of the drugs that were being developed weren't overcoming some of these properties. And so the concept here was take these tools and advance them and put them to use against targets that has some validity, at least from a biological component. So a really good example, it's back then in 2009, there was a new released software called Water Map, that was really to understand water molecules and how could we displace those water molecules with our ligands and really understand the protein ligand binding and the crucial role that water plays in the interactions. Nimbus' approach really at that point, Daniel, allowed us to be more highly efficient in drug discovery because now we're able to actually move faster, understand the fundamental molecular properties of the protein of target of interest, and designing a compound that would ultimately lead to a more efficient, say, of we're going to have less of a chance of properties emerging that are going to be more difficult to navigate as we get through toxicity and as we get to our development candidate and in hopes of filing an IND.
Jump forward to today, and computational chemistry is all around us. The ability to not only apply that, but to do so with large data sets and leverage AI has really changed what's possible. To what extent are you incorporating AI into your discovery platform today?
Yeah, and before we get to AI and ML and how we have been incorporating it, I think one fundamental component here at Nimbus has always been the tools and the platforms that we have are only extensions for our ability to go after targets. The first sin in drug discovery, as I have always believed, my predecessors at Nimbus have always believed, our investors believe, is target selection. So, without having the right target in mind, and the tools become ultimately useless, because at that point you're trying to validate the tool or the approach versus what you want to achieve by going after a said target. With that in mind, like most organizations, we have moved beyond just these physics-based approaches. Today, we integrate generative AI, machine learning-based AdMet prediction, retrosynthesis planning, real-time competitive intelligence, Daniel. I think one way to look at it is if I simplify some of the math around how much time it takes for a PhD lawyer to go through the patents that are being published by pharma, by biotechs across the globe, and you compare that to a tool that we have been developing internal to kind of go through patent literature to look at some of the structures to think about where we can get freedom to operate, we're talking about milliseconds and we're talking about costs in terms of you're not paying these prices for individuals a salary, you're actually doing this in cents of a dollar. And so there are various ways that we've thought about AI as being a force multiplier across various comp chem tools, competitive landscape, as I mentioned. But I also think, we're also applying caution, right? We can only deploy AI where it makes sense that it's faster and smarter. It's not just about automating everything. It's just really amplifying the conviction that we have to go with force against a said target of choice.
It's interesting you use that example of checking the patent landscape. I I think a lot of people look to very sexy applications of AI. In drug development, speed, cost, efficiency are so critical that I suspect that's where we're seeing at least the early benefits. But To what extent are you able to do things like find novel targets or explore novel chemical spaces because of AI?
Yeah. No, that's great. I think, you know, one way to look at this, if I talk about, your first part of the question, speed is only a part of it. I think the real win for us and our success of the compounds that we have discovered that are all novel, internally discovered, and the compounds that we've taken forward to the clinic, we've taken 4 assets to the clinic. It wasn't about speed. I wouldn't ever really feel comfortable saying Nimbus is the fastest biotech. I would say we have much more wins on precision, right? We're getting 2 better molecules. AI helps us design smarter, especially when we're pushing up against, tight selectivity or PK windows, even with some of the programs that we've had today. So, if I kind of take a look at where, the wins are coming from and the integration of AI, it is allowing us to scale at a faster rate, but also you have to have the right hypothesis to go after. I think we can expand our chemical space. I think novel targets, candidly, wouldn't be part of our hypothesis at Nimbus. There's probably more of a business conversation there to go after a novel target, to validate new biology. That's years, that's a lot of dollars at risk. And also just to evangelize a new target to the potential suitors, which in our minds are investors and pharma, would take quite a bit of validation. So for us, AI wasn't necessarily a tool to say, can we find novel targets? It was actually a question of, can we do 2 things? Can we improve on targets that already have some validation, be it genetically or biologically? And I think the other part of the picture from a Nimbus perspective is, can we use some of the tools to actually even kill programs even faster? And I think that's where some of the folks in our industry may be missing it. Maybe AI and ML have become a way to only go forward and succeed. But I think our definition of success is not just moving compounds forward, but also knowing when to stop. And the tools that we have been integrating have been helping us make decisions on, should we stop this program today, given the challenging chemical landscape, given the challenging clinical path, given the challenging commercial path to success. So, we're really trying our best to stay on the fence of not falling into the hype trap, but also utilizing the tools that we have today to really fundamentally increase our conviction and increase the hypotheses that we're testing.
It's not only about the tools, but the data, and I'm wondering, as you've evolved and the technology has evolved, are you using different types of data, greater volumes of data, and are you able to conduct more in silico experiments?
We are, and I think that's a big part of just as being students of the industry, being in the industry itself. We have a consortium that we work with across with CROs, we work with across with other biotechs. And the goal there is that for 16 years at Nimbus, we have generated a lot of data and a lot of programs that we're not going to advance. And so you're asking the right question. It's, can you take some of the high quality data sets and integrate those for free in exchange for all that data that we have contributing to additional training? And I think that is a big component of where I think a number of us in the industry are figuring out of how to preserve privacy, but enable these data sets. And I think that's such a unique part of our industry, that there's a willingness to share that information, a willingness to protect the privacy on the information, only to learn more. So the answer is yes. I think there is a good hearted effort to not only contribute to these larger data sets, but those larger data sets we are beneficiaries from. Candidly, Nimbus isn't producing all of the tools internally. So when our CROs are working with their own partners and generating these large data sets, we are also beneficiaries from that perspective as well. So I like that. I like the idea that, we're all kind of working for a greater cause, but yet in our own domains of how we're going to succeed by delivering for patients. Either I'm working on a competitive drug or a drug that deserves to get to patients, and yet also contributing to the greater good.
You alluded to targets earlier. Nimbus has been interested in using the technology to address targets that conventional approaches have struggled to succeed at being able to hit. How do you go about selecting targets, and to what extent is that defined by what the technology is good at doing?
Yeah. That's I'm laughing here because it's kind of been the perennial question externally. Even internally, we've evolved it so fast. When I started here 11 years ago, Daniel, you won't believe me. We had our board packs used to have, the new targets, and it's an evergreen list we have. And as we kind of go through a process to nominate the next 1 to 2 that we want to take into early discovery, and see if we can make progress and decide to move or not, we had this little kind of the precursor of, any kind of app to meet people. It was, is this target hot or is it not? And so as a BD person, when I started, it was like, hey, can you go and find out if this target's interesting to pharma? And it was always kind of a double-edged sword because we'd go to pharma partners and or investors and ask their opinion. And then you realize you're actually educating the world about potentially this target. So that I said, this is really not working out in our favor. And no one's really going to come back and say, is this target attractive or not when it's preclinical, unless you have competitive data you're going after. What I could say today, after being here for 11 years and the company being around for 16, target selection for us, I would say today now is a bit more sharper. It does start with clinical conviction. We ask, is this a target that matters to patients? And I know that sounds really motherhood and apple pie, but I think there's a lot of momentum that happens in our industry. And you being such a prominent voice in the industry, and you notice trends that people go and be trendy, right? It's obesity has a big play. Everyone runs towards obesity. If it's the IO space, people run towards IO. Nimbus was part of that narrative as well. And so I think, for us, we always try to figure out, let's start off with the fundamental question. You know, again, can we meaningfully improve outcomes if we get this target right? And that's always our North Star. And then we deliberately focus on areas of, I would say, unmet need, not as particular as a rare disease, but as a space of, we understand there's a challenge within the JAK stat space. There's a lot of black label drugs out there that are going after JAK1, JAK2, JAK3. Could we go after a target that could have incredible efficacy and overcome a safety limitation? That is what we would define as a breakthrough. And so we chose that target, for example, or the ones that we're working on now that have some hypotheses that have been tested, but they weren't successful because there were limitations either in the chemistry, there were limitations on the commercial landscape. But yet, for us, the commercial landscape was limited because you didn't have a good molecule to present. So we go back to our discovery engine and we go back to the fundamentals: computational chemistry, AI, translational design. Are we able to create an advantage here? And if the answer is no, we walk away. We also build differentiated platforms from day one, focusing on translational biomarkers, data rich phase ones. And if we can get all those to line up and continue to have the investor support that we have, it then becomes a question of, are we going to compete to be best in class or first in class? And I think once you get to those two questions, Daniel, you've probably already made a decision on, we're going to work on this target. And if we can't define clinical or commercial differentiation up front, we won't have the conviction and our investors won't have the conviction. And so we'll be able to pause that program and go back to the coffers and say, okay, what's the next set of targets that may be able to align across all those various principles?
I was going to ask you about whether this approach is yielding better candidates, but perhaps I should do so in the context of transitioning to the deals you've done to date. But what's the case that this does yield compelling candidates?
I would say we're getting better. I think, Nimbus's success has been purely pragmatism, which is led by a team that is cautious and aware about knowing how to refine and ask better questions. Our new targets group here, I think, is one of the most unique groups I've come across. Clearly, because I'm biased, but because the way we go about it, it's a very democratized approach to having all the scientists candidly bring their ideas to the table. You'll have a group of 15 to 20 scientists. Imagine entering, a Shark Tank version of new targets. and each one has a 20-minute pitch to cover rationale, genetics, biology, Nimbus's tractability, clinical path, competitive track, commercial path, and we'll go through an iterative process. And for whatever reason, that's allowed us to be sharper on the targets that we select, and the filter has gotten better. And so the targets that we have selected, they're competitive. They're on the list of the websites of Novartis, on Roche, on GSK, on Pfizer. And so we're not punching against, things that are, again, that need to be evangelized, that are novel. We're going against, you know, the big players out there that have conviction, and yet Nimbus comes out and says, we're going to create a better molecule. So I'm proud of the ability that we have selected the programs that we have selected. I'm more proud that we also know when to call it quits on certain targets. I've been speaking a lot about failing fast, and that's a big part of our success. KRAS was a target that we looked at 10 years ago, and we knew that the pockets that we were going after were yielding minimal results. We also knew that the work that was being done by Amgen and Mirati most likely would yield success for patients. And that's probably the right thing to do. So Nimbus would say, let's pause that program, find the next target, and invest in the ROI there, to knowing that we can probably go towards another space, another therapeutic space even, and have success. So I think the target selection process, it's still evergreen. We're going through it now. We have a list of targets that we're prosecuting that are undisclosed publicly. We have to be aware of the competitive environment here and globally. And so that has never changed at Nimbus. I think that's a big part of every round of financing we go to. There's always a big dedicated amount of capital to continue this idea of select and find the next set of targets, ask the right questions, invest early, and kill early as well.
You talk about failing fast, which is a mantra in this industry, but often for cultural reasons, difficult to do. Why do you think you're able to do that? And is it a matter of size or other issues that allow you to recognize quickly that a program isn't worth taking forward?
Yeah, no, I think you said it yourself pretty well. We've been really fortunate to cultivate that level of culture. This is ultimately, of course, every organization has structural leadership. The difference I've always felt at Nimbus is that every one of us is an owner of the company, not just speaking from a pure equity perspective. This is their company as well. And so individual scientists at whatever level it may be, from senior director, executive director to VPs to SVPs, all have a chance to nominate a target that will shape the future of our pipeline, the future of our company. And equally, that level of maturity and responsibility allows them and empowers them to say, this may not be the program worth pursuing. And again, I don't know how we got fortunate to have that culture. We celebrate all of our wins and we celebrate all of our failures. We celebrate when a project team is asked to make the decision to pause a program. And we keep a high bar, Daniel. We keep a high bar on what's a go, no go. We've had programs we talked about in the past that we were going after an immunology indication, and the therapeutic window was way too narrow, that if we were any other company and the ego was driving us to say, let's validate that pathway, let's validate and convince ourselves it's the right target, we will go after an oncological or a hematological indication. The bar of success here says, you know what, let's not compromise with our standards and move forward. And I really, really attribute that success to years of really investing in our team, years of building a culture of leadership, years of building a culture of authenticity, of asking people to share what their own hesitation might be, asking them to be objective, but yet have conviction. And I think if I were to summarize it for you, the culture at Nimbus has both. We have deep, deep conviction on the programs we're working on, and we also emphasize deep discipline, because if we're not going to make progress, then we're probably foregoing another program which would be equally or more beneficial for patients. And that is my style of leadership. That's my own perspective to bring back to our team of PhD science level theses that we're writing on why we should have conviction, but now I'm asking them to be operators and managers to also apply that discipline. And so far, I'm very fortunate to have a team that reciprocates that level of care. To be able to say, you know what, we're probably going to pause this program, and there may be a suitor in the future who may want to pick this up at that right point.
Well, let's talk about one of those wins, which I think makes as good a case as anything for your approach. In 2023, Takeda acquired an experimental TYK2 inhibitor from you for psoriasis and other autoimmune diseases for $4 billion upfront and the potential for two additional $1 billion milestone payments. It was an eye popping deal, particularly considering it wasn't the first TYK2 inhibitor. What was unique about the Nimbus molecule?
A couple of things stand out. We were going after TIC2 for quite a while. There was deep conviction from an immunology side of how this could potentially overcome some of the safety overhangs from the Pfizer's and the Jackstats place, and others that had black box warnings. Bristol was also pursuing a TIC2 inhibitor, and some call it a Jack 4. For whatever reason, this fourth stepchild was named differently. So TIC2 is the name. And for us, Daniel, to your question, this was a case study for Nimbus in precision design. BMS's deucravacitinib, which is now commercially available as SOTIC2, is a strong first-in-class molecule. Our goal was purely differentiation on best in class. And so through the computational approaches that we had, I think the first thing that stuck out here, Daniel, was our TIC2 molecule achieved 1.5 million fold selectivity over the other JAKs, so JAK1, JAK2, and JAK3, compared to the 75x that Bristol had for deucravacitinib. And that goes back to structure-based drug design. We were at a molecular level to understand the interactions with cysteine residues that Ducra had that we were not seeing. And we were able to push our molecule to such a high level that we were not hitting any of the other JAK families, which in our hypothesis would overcome any safety. So safety was in the conversation, and that became efficacy. And as we ran through our two phase 2b studies in psoriasis and psoriatic arthritis to get to an efficacy bar that was closer to the biologics and far superior than Ducra is ultimately where it began an extremely competitive process with multiple partners that saw differentiation engineered through Nimbus's platform, and ultimately, to your point, was core to the $6 billion partnership with Takeda. And in Takeda's perspective, not only was psoriasis and psoriatic arthritis a near-term catalyst for potential revenue, it was an ability to say, we can take a molecule with such a clean profile and go into potentially 9 more indications. So the amount of capital they're going to commit to go after other immunology indications is clearly what a small biotech like Nimbus couldn't afford to do. But what we could do is use our fundamental conviction and our fundamental computational chemistry tools to really derive and create a differentiated molecule that ultimately played out into the clinic and successfully.
You also did an earlier deal with Gilead in 2016 for $400 million up front and up to an additional 800 in milestones. This was for an experimental therapy for the liver disease NASH, which at the time was without an approved therapy. What does this say about your discovery approach?
The ACC program, again, was another molecule or another target we were going after that had been around, 20 years. Pfizer, others had made progress, but they weren't able to get into certain pockets with molecules that were effective. ACC, our drug is now called Firsocostat in Gilead's hands, was originally being conceived for diabetes. As we started putting it into various preclinical models, we started seeing impact on inflammation, started seeing impact on steatohepatitis, um, and we started seeing impact on fibrosis. And at that point in 2013, No one really talked about NASH. We were talking about NASH, and everyone said, "What is this liver disease?" Not really well known until 2014. Intercept had their phase 2b results, and the word NASH became almost common household. For us, then the hypothesis was push this further into phase 1b. And the program showed we could take tough, metabolically complex target and turn it into a viable selective molecule that can go deep into the clinic. What it showed us, to your question, Daniel, is that it validated our discovery engine and gave us one of the most meaningful early BD wins in Nimbus's history. It wasn't just the financials and the milestones that we've achieved to date. And we're hoping that, Gilead is successful, but also recognizing the world of MASH has evolved dramatically with the GLP-1s coming into space. For Nimbus, it was a validation of target selection, validation of we chose the right computational chemistry tools, validation of we're being open and thinking through what's the clinical path to success look like. We're thinking through commercial success. So that first BD win wasn't just a significant return of capital for investors and employees. It was a significant validation of, okay, go back to your confidence of selecting targets, go back to your confidence of your fundamental belief in computational chemistry, go back to your confidence in how can you navigate and build an efficient team to de-risk the program and get through the clinic.
We've talked about your innovative technology and your innovative approach to drug discovery, but I'd argue your business model was also fairly innovative at the time. You're an early example of a hub and spoke approach. You set up separate LLCs for your assets and use a lot of external talent. This is capital efficient, which is good for today's financing environment. But can you talk about some of the other benefits of this approach in terms of providing liquidity to investors and making it easier to structure deals with potential acquirers?
Sure. This is a topic that I love to get in depth to, a topic that I think our lawyers have made quite a bit of money on giving advice on the LLC structure as well. I think if I step back, why was this put together? Back in 2008 and 9, when the Nimbus business model was being derived, it was about how do you protect a company? I think it starts from there, right? If you have a platform, but you don't have any necessarily residual IP related to that platform, can you fundamentally separate the asset? And in all my relationships with all my pharma colleagues, if anyone were to approach us from an M&A perspective, I usually kind of ask more questions than anything else about why do you want to acquire us? Do you want to acquire us because you want to get rid of a competitor? Do you want to acquire us because you want to create an R&D footprint in the East Coast? Do you want to take our team members who are highly talented and integrate them? Or do you want the single asset? Or do you want the platform? And the answer, as you know, they want a single asset. So how do we create this business model that returns capital in perpetuity? And so each of the assets, and I'm going to be clear, the word asset, the word subcorporation, the word target are pretty synonymous at Nimbus. So if I say Tick2 by itself is a subcorporation that has a name. It was called Nimbus Lakshmi. And all the surrounding compounds and all the data related to anything Nimbus has ever touched within the Tick2 field would be sitting in that subcorp. And that allows us to partner or exit them very independently. So structuring the deal from an M&A perspective is probably the easiest. They essentially get to buy out a whole subcorporation. And the LLC works its way through the beautiful world of tax efficiency where they buy out the LLC, capital gets returned to investors because every investor and every employee at that point is treated like a partner. So you only pay tax one time. So we always kind of give financial advice to our colleagues of no one go on a buying spree right now. Wait to pay your taxes, and then you'll know how much net benefit you've made from a deal. And that has truly rewarded us in a way of we've done deals, if you combine our M&A deals and our licensing deals, north of 8.5 billion. We've returned $4.1 billion in capital back to investors. So that's the upfront capital that we talked about from the Gilead deal, the upfront capital minus all the expenses and obligations we had paid from the Takeda deal. So 4.1 billion gets recycled back. And it gives our investors, candidly, a lot of flexibility. They get early liquidity or liquidity, and they get to decide if they want to come back and finance Nimbus again. And so we don't have a traditional model where it's called Series A, Series B, Series D. We do it by year. So Series 2023 was the last round we raised because we reset our cap table. We gave everybody their return. And once they have their return, they can decide to come in the next round, or we can go out and raise money from new investors who have come in as well. So that flexibility allows us at Nimbus to, on paper, operate like a investment firm if you would have, right? We're thinking through how does our salt-induced kinase program today look like compared to our WRN program, look like compared to the early discovery program, but if and when you come to our offices, it feels like a normal company. We're working across a horizontal portfolio. We're all working and interested on what's happening on Warner, on SICK, and really managing a team that's excited about the portfolio. But when it comes to transactions, when it comes to partners, they're very much focused on a single asset and thinking about a deal construct from that perspective.
It's interesting because the one thing it doesn't do is free you from the capital markets. The company is private. It raised an impressive $210 million round that was co-led by GV, the former Google Ventures, in 2023. How is that money being used, and how far will that take you?
Yeah. So if I step back one second, we are fortunate to have remained private after 16 years. And one of the reasons we've been able to do that, Daniel, we don't look at the IPO as the exit. It's a way for us to get financing. And we could say that because from an investor's perspective, they have to and do believe that Nimbus's exit is going to be in a form of a business development relationship, as we've always done in the past. And so the $210 million that we raised, and as you had mentioned, Google Ventures, Bain, RA Capital, a number of other investors from the past as well, Atlas, SR1, Lightstone, SV Health investors, continue to believe that the model at Nimbus is going to have a BD perspective to it. And when we raised this capital, it was around our two lead programs. One we just launched in the clinic is our Warner helicase, a synthetic lethal target for patients that have microsatellite instability solid tumors. These are cancer cells, and these cells um, have a survival mechanism by having this helicase continue to keep that cell alive and functioning forward. We have a second program now in immunology. It's a salt-induced kinase 2 program. It's been looked at and studied for years. Academically, even pharmaceuticals, Galapagos tried its best to get a purely selective program. That's Nimbus' specialty, using all the tools we've talked about. And we have a SIK2 isoform selective compound moving forward. We're going to get to IND early next year and looking at first in human next year, end of next year. And the rest of it scaling the translational and clinical operations. And as I mentioned before, it really allows us to think about the future of our portfolio and the early drug discovery. It gives us enough runway to hit major data readouts and potential partnerships without having to return to the market in the near term. In this capital environments, um, I count my blessings to be able to say we have capital to take us through these milestones all the way through next year and actually even out into 2027. And that doesn't mean we're going to sit back and not do BD. There's early targets that haven't been publicly disclosed that pharma is engaging, and they're engaging with us because knowing that we have an ability to select winners, we have an ability to most candidly kill programs, and we have an ability to think through how to structure deals that preserves rights for Nimbus, that preserves potential upside for investors, but most importantly, has an ability to say, we're happy to partner with the right partner on a program, knowing that we have a belief in our Warner program and SIG program and others that may ultimately provide upside for investors, but most importantly, be beneficial for patients.
Where do you think we'll be in the next 5 to 10 years with AI in drug discovery and drug development? And how significant an impact do you expect it to have on that? And to what extent do you think it'll change what's possible?
I think the conversation last year, Daniel, was the perennial, is this hype or reality? We've moved past that at Nimbus. It didn't help us to be folks on the sideline pondering, is this real or not? For us, it was very much real, and we moved forward exactly how we move forward by embedding computational chemistry and taking it to the next level. The 5 to 10 year window, it has to be embedded at most organizations. I think it'll be embedded in every step. No one will talk about AI biotechs. They'll just talk about smarter biotechs and the way you integrate them. We still will apply caution by not falling into the hype trap. We still will apply caution by not over-promising speed, by over-promising revolution. I think the ones who will win will be the ones asking better questions. You won't succeed by predicting everything, but you will succeed by designing good experiments. You will succeed by choosing the right targets. You will exceed by managing the portfolio. And so that is where I think there's a big delineation between individual organizations, that AI is going to be a multiplier, but not a revolution or a replacement. So I'm hoping, and what we are seeing really quickly, is that the companies who have already begun integrating the AI narrative are the ones who are still talking about the assets and the quality of compounds versus the ones who are talking about the platform or talking about the AI revolution. And so it's a very unfortunate time in the market. I do believe the bar is higher, and that's a great thing for patients. The bar should be higher for the quality of data. And it's also a great time for people to begin this integration process, because going back to the 5 to 10 year window, we're all going to be integrated. We're going to have these tools available. But that does not mean you're replacing the talent that we have, the medicinal chemist, the computational chemist, the ones who ultimately have to make the human judgment on, will this molecule be used in QD or BID? They may have the data sets to help make the predictions, but those calls will ultimately come back down to the team and the winning teams that actually make a decision to make this molecule the most impactful for a said disease.
Abbas Kazimi, CEO of Nimbus Therapeutics. Abbas, thanks so much for your time today.
Daniel, what a pleasure.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.